It's on.
Test, test. Oh, good morning, everybody. Welcome to Dr. Hugh Ross's Paradoxes Sunday School class, and he's away from us today, so we have a couple special speakers for you. Today is the 28th of May, 2023, and today we're going to hear from Ross Hoagland. He's our co-coordinator here for Paradoxes. He's a longtime apologist for Reasons to Believe. And Tim Wagner. So they're going to be speaking on the origin of life on Earth. Is there a natural process that can explain it? So we'll have, at the end of the talk, we'll have a Q&A period. So be ready for that. And come on up, guys. And while you're up here, I'll say a quick prayer to open us up. Lord God, we thank you for this Sunday and just ask your blessings on our presentation today. Open our minds and hearts, Lord. Prepare us for using this discussion today. Bless the speakers. Bless Hugh Ross wherever he is, Lord. And we thank you for our country. Thank you for tomorrow's holiday, Memorial Day, for those who died in action, for our democracy and peace. Thank you for all this in Jesus' name. Amen.
Amen.
Okay, guys.
Thank you, Mark.
Thanks, Mark.
Hi, everybody.
Hello.
Greetings. Welcome to Paradox class. I'm so excited because Tim Wagner's up here with me. We're going to talk about the origin of life. It's one of the toughest, most intractable problems in science. And the question on the table is there a natural process that can explain the origin of life? Just say what you're saying, Jamie. You want us to bring it over? Okay, good. I'm going to tell a little story. I've always loved science ever since I was a little kid. I wanted to understand how things worked and how stuff put together. And when I was a kid, I didn't have any tools, but I wanted to understand things. My dad gave me a sewing machine to take apart. I took it up on the roof and I threw it off the roof onto the driveway because then it busted open and I could try to understand all the little mechanical stuff. So I've always loved biology. I've always loved the beauty of biology. A couple of years ago, I was out in my little front yard and I had a cup of coffee early in the morning and I was looking at this really cool tree that we've got, and I was kind of starting to thank the Lord for this tree. And then I started to think about all the processes that go on in that tree. I don't have a degree in biology, but I've studied it enough to really understand how unbelievably complex it is. And I had a little God moment there. I really did. It was more than just appreciating the tree. It was appreciating God for all those intricate, amazing systems that go on in biology. A lot of this we didn't even know about 100 years ago. It's been there the whole time, and now we're figuring it out. So that's kind of where I'm at this morning. I've always been interested in what is that very first life that came into the earth. How did God do that, or how was it created naturally, if you believe that? So, Tim, do you want to say something about your
Sure,
story?
Yeah. First of all, it's great to be here. Thanks for having me, Ross and everybody. I feel a little intimidated because most of you all are more well-read than I am, more intelligent, but I just come here as a student to share what I've learned. I recently got my degree in organismal biology from APU, and I went in at the organismal level, about, body plans, body structure, stuff like that. And as I took more classes, more of the advanced classes, I fell in love with the molecular side of things, the small things. And going into that, I came out with an amazing appreciation for how beautiful, how sophisticated the machinery is within the cell, and that it's almost incomprehensible, and that I could probably spend the rest of my life learning about it. There is just so much to learn about. And a lot of the stuff, like Ross said, photosynthesis, we didn't know how that worked 100 years ago. A lot of the molecular stuff we didn't discover until 10 years ago, until a few years ago. And so it's exciting to learn about molecular biology and how it's one of the coolest frontiers of science, in my opinion.
So secular science tells us that the origin of life was a completely undirected natural process. What we want to do in this talk this morning is talk about what would that actually take? Is that a reasonable hypothesis? What would it take for a natural process, an undirected, mindless process, to produce the first life that came into existence on planet Earth? So let's talk a little bit about what are we talking about when we talk about first life? What did it look like? Well, we know that first life came on the Earth about 3.8 billion years ago. Not much time after the early Earth cooled enough for life to even exist at all. It was a small, simple, one-celled organism like a microbe. We would call it a bacteria. And one of the things that I want you to understand is we're talking about something that is utterly complex and yet can't be seen with the human eye. There's so much going on in that tiny little speck that you have to see under a microscope. You can't assume that it's simple. So in this talk, we're going to talk about, is it possible that this came about naturally? Why should we care as Christians? What difference does this make? We want to make the case that single-cell first life is way too complex, way too elegant for any natural system to produce it. And the beauty of it is better understood if we posit a designer who created it intentionally. So we want to start with a sample of some articles from popular literature that talk about origin of life. As we go through these couple of quotes, think in your mind, are they making it sound simple or are they making it sound complex? Tim, why don't you talk about these?
So our first quote was from a magazine called The Conversation, and they had an article that said, "These simple molecules," talking about the building blocks, "combine to form more complex ones, such as fats, proteins, or nucleic acids," that's DNA. "Once an information system can make copies of itself, natural selection kicks in." We also pulled a quote from a scientific paper, and this one, the researchers say, "We believe the change was sparked by a few small precursor proteins that perform key steps in an ancient metabolic reaction, making them a plausible starting point for life."
So the question we want to ask is it really this simple? Naturalists make it sound not only simple, but almost solved, right, from those quotes. What I asked Tim to do is put together a short little primer on what that first bacteria looked like. So Tim?
So, what was first life like? As Ross mentioned, most researchers in the origin of life agree this was most similar to a bacteria. A bacteria is a single-celled organism. Of all the organisms on planet Earth, it's the most, quote-unquote, simple. It doesn't have organelles. It doesn't have a nucleus to house its DNA. It has one chromosome that's a circle, unlike ours, that is linear. And now, we're going to assume that this first life form was as simple as possible with the smallest genome possible. It didn't start with an enormous genome or anything. Now, the smallest autonomously replicating life form, the life form that can duplicate on its own, is a sexually transmitted disease called Mycoplasma genitalium that lives on, you guessed it, your genitals. And this life form is the smallest genome on planet Earth with 1 million base pairs, slightly over that. And in 2016, a group of researchers wanted to design the most minimal, the smallest genome possible for bacteria. And so they started with this candidate, which is a great candidate because it already has the smallest genome. And what they did is they introduced mutations to random genes and knocked out random genes until they could reduce it and still have it replicate on glucose. So it was given plenty of food. But other than that, there were no other life forms present. So it's not technically a parasite in that sense. They cut it all the way down to 531,000 base pairs. But after this is about 500 genes. After you cut it down further than this, it can't replicate anymore. And already this thing is missing a lot of important genes that would give it more survivability in a more diverse environment. Keep in mind, this is in a lab with plenty of glucose to feed it, right? So they cut it down to about 500,000 base pairs, about 500,000. Now this first life, whether it used DNA or RNA as its genetic material, would still need 3 parts to make up that genetic material. You need the nitrogenous bases. You might have heard of this, A, T, C, and G, right? The 4 letters that make up our DNA.
That's like code. It's like a written alphabet in a organism, right?
Exactly. Instead of like zeros and ones like binary, it would be 1, 2, 3, 4, right? But in addition to these bases, you also need 2 other very important molecules. You need phosphate, which is phosphorus with 4 oxygen atoms bonded to it, and ribose. This is even more important. You get phosphate from rocks, but ribose is a 5-carbon sugar, and this has no known natural generation. This is basically only created by life forms. This needs to be present because it forms the backbone of the DNA, in which the nucleotide bases reside. It gives the DNA structure. Not only this, this first life form would have had a cell membrane made of a phospholipid layer. Phospholipids generate the cell membrane that every organism on planet Earth has. This is what separates the inside of the cell from the outside. The phospholipids are water insoluble. So what they do is they create an impermeable membrane. And so with proteins embedded in there, which I'll get to later, the cell can control what's inside and what's outside. Without this, it's just floating around in an environment ready to be destroyed. And finally, and I would say this is maybe the most overlooked part of first life, the average bacteria is about a cubic micron. And in a cubic micron, you have 2 to 4 million proteins floating around, performing all of the processes needed for the cell, all the work that needs to be done, all the complicated physical changes, steps that need to be made. These are made by the proteins. I want to emphasize this. This is a really important point. Proteins are just as important as the genetic material. People often think of life as, oh, it's just DNA, and from that, as we saw in a previous quote, once the DNA's in a certain sequence, it'll just make copies of itself. This is not possible. Without proteins, DNA is not life. And proteins without DNA is not life either. You need both. So, what are proteins? Proteins, like nucleic acids, DNA, it's a long chain. But instead of 4 bases, now you have 20. It's like having 20 different characters. And these amino acids, the 20 different amino acids, are all joined by what's called a peptide bond, as you see on the right side of the image. Now, I want you to remember, this is a special type of reaction. It's called the dehydration synthesis reaction. You don't need to remember that part, but just remember that a water molecule is kicked out when these 2 join. You see the OH from one group and the H from the other combine to make H2O. The water molecule then exits, and the 2 form a peptide bond. Now remember, this is an endergonic reaction. What that means is energy is needed to be input into the system for this to happen. It's harder for the bond to be made than it is for the amino acids just to remain separate. So we need energy to be put into the system. Remember that. But that's not all proteins are. They aren't just long chains. They have very specific structure, and the structure is what makes them function. Ross here has a belt. He'll follow along with me. So the primary structure of a protein, that's what I mentioned before. That's just the sequence, right? 20 amino acids all put in a certain sequence. That's its primary structure. But that alone is not enough to give it function. You have other forms of structure. Secondary structure. Now, depending on what amino acids are put in an order, the long chain of amino acids will then spontaneously fold into certain structures, things like alpha helices, spirals, or beta pleated sheets, when it goes back and forth and creates a rigid wall. These are very important for function later on, but this is not everything. After this happens, it now forms what is called tertiary structure. This is, for some proteins, the final form of structure, and it can't do this spontaneously. This doesn't happen on its own. It needs other proteins already made and folded to come in and help it fold to the correct formation. Otherwise, it'll fold randomly and will never fulfill its structure. So you need proteins already in place to let proteins actually function as themselves. And then finally, many proteins exhibit the fourth level, quaternary structure. This is where multiple proteins already folded in tertiary come together and aggregate to form one big multi-protein conglomerate. So what are these proteins needed for? I don't have time to go into all of these things, but I just wanted to show you, this is a truncated list, all right? This is simplified. I left out a lot of things that this early life form might not have needed, like it might use ribose, the five carbon sugar in its DNA, as its sugar. Maybe it didn't need glucose. So I left out all the glucose-related stuff out of this. And I'm just going to go through a couple of the important ones. And the first one you see in the top is DNA repair. And a lot of people don't know this, but there are more than half a dozen different repair mechanisms that are constantly whirring away in your body, repairing your DNA. When your DNA replicates, anytime your cell divides, it has to make a copy of its DNA. And every time it does that, it introduces thousands, if not tens of thousands of mistakes. Now you have all these other systems, which are proteins, coming in and literally cutting out bases, replacing them with the proper ones, or having the cellular machinery go back, take the one out, put it back. You have other ones where if the whole strand breaks, you have proteins that come in and put the entire strand back together. These are really necessary. Without these, we would quickly mutate and die within a matter of days. There's a bunch of others I won't go into, protein folding as I mentioned before, but I'm going to skip to the end. We have third to last membrane transport. As I mentioned before, there's a phospholipid bilayer, right, around every organism, and it's impermeable to water. But if there's no channels through it made by proteins, the cell inside is going to suffocate in its own skin. It won't get nutrients back and forth. So in reality, you don't just have a phospholipid bilayer, you have one that's studded with millions of proteins, some channels, some receptors that take in and spit out nutrients and water and all that stuff in order for the cell to survive. Second to last, we have ribosome biogenesis. Now, ribosomes, without them, life can't exist. So, I'm going to go into that really quickly. What are ribosomes? So I have here on the screen on the left side, that giant multicolored blob is a bacterial ribosome. And on the right is a ribosome that's in our cells. Now the ribosome is what assembles the proteins. It's what takes the amino acids and starts forming those peptide bonds. Without this really super complicated structure, those peptide bonds won't form. This protein is actually 60 different proteins, the bacterial one. 60 different proteins from 60 different genes all coming together. There's also 2 pieces of ribosomal RNA, which I won't get into, that give it additional structure and function in there. This thing is incredibly complex. And in any bacterial cell, there's tens to hundreds of thousands of these whirring away all the time, making the proteins necessary for this thing to survive. This guy, the bacterial ribosome, is coded by over 20,000 nucleotide bases. So in our 500,000 base pair genome, that's already a huge chunk goes to just this ribosome. And the ribosome biogenesis is only a fraction of all the genes needed. Now after that is DNA replication. Of course, we all know that DNA needs to be copied. It needs to make 2 copies if a cell wants to divide. But in that DNA replication, there's so many proteins involved. Like I mentioned before, proofreading, DNA repair, but also there's many proteins involved in this process, and I think a video will do it more justice. So I have a quick video I want to show you all.
If we unwind two new DNA molecules. Now, you have billions of this machine right now working away inside and copying your DNA with exquisite fidelity. It's an accurate representation, and it's pretty much at the correct speed for what's occurring inside you. But I left out error correction and a bunch of other things. This was worked from a number of years ago. Thank you.
So he said there at the end, he left out error correction. That's actually the simplified version. In reality, there would be all these thousands of other proteins all coming together to repair it, to bring in more amino acids. And also, this thing, it looked like it was just floating in a vacuum. In reality, it would be in a densely packed soup of other molecules and proteins that this cell is using. So hopefully that gives you a little bit of appreciation for some of the machines that are whirring away inside your cells, as he said. These proteins are incredibly complex. Now this image here, this is a computer-generated image. The colors are put in there for you to distinguish the parts for yourself. It's not how it actually looks. And this is not a bacteria. This is one of our cells. It's actually a very small fraction of one of our cells. I don't know if you can see the smooth white line on the right is the cell membrane. And the little tiny one on the left, on the even more to the right, is the cell membrane of another cell. And just looking at this, you can see here on the left side, that what looks like a double layer inside of there, that's all the genetic material, all that blue stuff. And those giant gates are really complicated gates that basically allow RNA to exit the nucleus and enter the cytoplasm. Basically, it's regulating what's inside of the nucleus and what's outside. So just looking at this, you can see there's so much going on. And a bacterial cell would be smaller, but I would say it would have just as much density and complexity as this.
So you're hearing a lot of different things. I hope you're like overwhelmed with the beauty and the complexity of what you're seeing. And yet you read these scientific papers or you read science and they talk about simple life, right? Well, which is it? How do we think about it? So this next slide is really important. How does the complexity of life compare to man-made objects? Okay, so you get a sense of what we're talking about. Okay, think about some stuff that we're all familiar with. Let's start with a hammer. A hammer is a single moving part, one part, right? Then we get a little bit more complex and we have a pair of pliers. A pair of pliers has a pivot in the middle and it's two moving parts. Then we go to an electric drill. Electric drill introduces now, it's not just mechanical, but it's also, there's electrical circuitry in there. Now we go to a laptop. Think of an old school laptop that had a hard drive that spun. So you have moving parts, you have electrical parts, and now you introduce information. Next one up is a car. A car is a great combination of a bunch of moving parts, sophisticated electrical systems, and high-tech electronics. The next step up would be a whole collection of these systems that make up a human city. So from simple to complex, you start with one part, two parts, hundreds of parts, thousands of parts, tens of thousands of parts in a car, and millions of parts in a city. Okay, now let's look at the next scale.
So I picked some organisms for our organism complexity arrow. And at the start, I have bacteria. I didn't put viruses. Viruses are simpler and smaller than bacteria, but many biologists argue whether or not viruses are life. Since they can't replicate on their own, if we use that as the definition of life, I would say they aren't life forms at all. They need to hijack other cells' complicated machinery. So I would say they don't count. So we're going to start with bacteria, right? Single cell, no nucleus, one chromosome that's a circle.
Just like what we found on Earth, just like what the first life form on Earth would have been
Exactly.
like.
And then next you have a yeast cell. Now we're going to a eukaryote. This means it has a nucleus, it has organelles, but it's still a single cell. It's much more complicated though. After that you have a sponge. Now it's multicellular, right? Many hundreds, if not millions of cells all coming together, but very simple body plan. Some pores to allow water to come in and out, but that's pretty much it. And then you have an oak tree. You might say, oh, oak tree, is that really that much more complicated than a sponge? Yes. It has many systems in there, the xylem and the phloem to deliver water and nutrients up the trunk, a root system that reaches out and makes contact with the soil, but also makes contact with bacteria and fungi living in the soil to help give it more nutrients. And it has leaves that photosynthesize with very complex machinery we couldn't get into, but also has stomata, these holes that open and close that allow gas exchange into the plant. So already a plant is very sophisticated. And after that, you have an animal. And I put a human because I think a human is the most sophisticated of all creatures. But with any given animal, you now have motor systems, you have a digestive system, you have a nervous system, all using even more complicated machinery than a tree, despite having maybe fewer atoms.
So here's the question. How do those two scales relate to each other? Okay, if it's simpler on the left and more complex on the right, we understand the man-made objects pretty well. We're all familiar with every one of those. The complexity in a bacteria is really unknown to us. So what I want you to do is get this in your head. Look at it straight up and down. Is a bacteria about as simple as a hammer? No, way more complicated. Is it about as simple as an electric drill? No, way more complicated. In fact, a simple, there's that word, single-cell organism, just like the very first life form that appeared on Earth, is about as complex as a human city. There's transportation systems, there's waste systems, there's electrical systems, there's error correction systems. It feeds, it spits out waste. It is insanely complex. So when you hear these terms in the popular literature about simple life, look back on this and remember, what does simple mean? It's simple compared to those other biological systems, compared to a yeast cell, sponge, an oak tree, or us. But when you compare it to human systems, it's about you think about it in terms of about the complexity of a city. Okay, we have one more analogy that we want to use to talk about how difficult it might be for this single-cell organism to come into existence by itself. We want, in the next few slides, to ask yourself, can this car be assembled? Okay, what's the next slide, Tim? We have our friendly auto mechanic here. And what has he got? Well, first of all, he is in a controlled lab that you can't see, but just assume he's in a building that has no outside influence. Controlled temperature and lighting. He's got a set of plans in his hand to build the car. He's got tools in his hand to put the car together, and he has all the car parts strewn in front of him that will build that car. Again, what's the question? Can he build the car? Okay, what's the next slide? What if we take away his controlled environment? Now he's got the plans and the tools and the car parts, but he's outdoors in a chaotic environment similar to what the early Earth might have been. Okay, what's the next step, you might guess? Now he has no plans. Notice the X on his chest, right? Okay, so he has no controlled environment and no plans. He still has the parts, and he still has the tools, and he's still a pretty smart auto mechanic. Can he build the car? What's the next step? He just lost his toolkit. Now he has no controlled environment, no blueprint, no tools, but he still has the parts, and he's still a pretty smart guy. Can he build the car? What's the next one? The parts went away. The parts became what we made them from, right? Dirt and sand and mud and water. So now he has no controlled environment, no blueprint, no tools, no parts. Can he build the car? And what happens next? No mechanic! There's no controlled environment, no blueprint, no tools, no parts, no mechanic. Can that car get built from just dust and dirt and mud? That's what we're looking at, folks. That's what a naturalist will tell you, that life came into existence from that. Tim, what's the next step on there?
So you might be wondering, are these truly analogous to how organisms work? And I would say, yeah, they're pretty close. The no-controlled environment would have been like the chaotic early Earth. We're talking about radiation from the sun, UV that will disrupt genes. We're talking about storms, wind, a mixing environment, all this sort of stuff that would make it really hard for life to form. But not only that, no blueprint. We're talking about no genome to start with. There's no genetic material to start with. So there's no blueprint to make the proteins in the first place. There's also no tools. This is like not having the proteins. As I mentioned before, the proteins are the machines. Those are the tools that do the stuff in the cell. Without those, even if you have the whole genome sitting there, it's just going to sit there. It's not going to do anything. And then finally, of course, if this is coming about through natural... Oh, sorry, I missed no building blocks. Sorry. A lot of people assume there would be all these amino acids, all these precursors already there, ready to be assembled. But as you saw in our analogy, it's more analogous to having just dirt. Try to make the car from dirt. Because a lot of these precursor amino acids, nucleotides, they didn't exist. They're really hard to make through natural processes. And finally, of course, we're assuming there's no mind, there's no designer, there's no God. This is just wind blowing about by chance. Can this cell be assembled? The simplest cell possible.
So take the analogy further. What we have in a car, average car nowadays has about 30,000 parts, right? Well, in this picture is about 30,000 grains of rice. So what we're talking about with a car self-assembling is about that many grains of rice. But in a bacteria like the first life that came into existence, you're talking about 1 million parts.
More than 1 million.
More than 1 million. This is about 1 million grains of rice. Okay?
No, not yet. That's 500,000.
Oh, there we go.
All right. That's about 1 million grains of rice. So I would need 99 more bags of this to show you how many parts are in the simplest bacteria possible. Around 100 million. It'll probably fill this room, right?
So that's what the naturalist is up against. The naturalist, the person who believes that first life came into existence in some kind of undirected natural process, has to explain how 100 of those could have self-arranged with no known natural process that could take care of that.
But now there are actually other problems we face for a naturalistic origin of life. We're talking about there's 2 issues of time. The first is the assembly time. The genetic material for this organism would have arisen entirely within hours, because within even minutes, UV light starts to damage the DNA, and within hours, this thing's already going to be so damaged, it's not going to be functional. So we're going to need this entire genome, 500,000 base pairs, to come in together within a number of hours. But not only that, we have a small geologic window for this to happen. We know that there was only about 5 million years between when Earth was cool enough for liquid water to form and life popped up. You might say, oh, 5 million years, that's a long time.
And it could have been way less than that.
Could have been less than that, yeah. But 5 million years in the perspective of Earth is actually the blink of an eye. So you don't have billions of years. You have 5 million at most. That's not the only issue. These things can't happen anywhere on Earth. Now, if you remember back to the peptide bonds I was talking about, water is kicked out, right? One of the biggest problems to having the oceans, to having water as the starting point of life, is that just the way thermodynamics and kinetics work in the presence of bulk water, that peptide bond is prohibited. So basically, there's pressure to stay separate amino acids in the presence of water. So having water, we always thought that must be the starting point of life. Turns out proteins can't even form in water spontaneously. They need proteins that are already made. That's not it. So you say, okay, what if we're out of water? What if we're in aerosols floating around in the wind? Well, as I mentioned before, light is very destructive to the genetic material. It forms what are called pyrimidine dimers. This is where 2 types of bases will stick together and form a kink in the DNA. This will interrupt any cellular machinery chugging along trying to replicate it. It's going to stutter on that bump, and it's going to mess up. And within minutes, you have thousands of these happening to any single genome. And finally, you need temperature. You need high temperature because, as I mentioned before, that's an endergonic reaction, meaning energy needs to be put into the system. It's got to come from somewhere, and the most likely place is heat. Problem is, you can't be too hot, or else you start breaking down the molecules you just started building, which, as we realize now, it's really hard to even get them started in the first place. But that's not the only issue. This last issue, the issue of chirality, is often overlooked even by origin of life researchers. So what is chirality? Chirality, or a chiral molecule, is a molecule that has a mirror image. And its mirror image is not identical to what it is. So Ross here can demonstrate with this pair of gloves.
So we're all familiar with this. It's a pair of gloves, left-handed, right-handed. They're identical, right? They have made out of the same material. If you turn one, it has the same outline. Same material, same size, same weight, but they're completely different. I can't put the left-handed glove on my right hand, right? It doesn't work. This is a really good example of a chiral system. And what Tim is saying is that there are, in nature, some of these molecules are made in both left and right-handed forms. And so that early first bacteria that was made would have had left-handed and right-handed molecules sitting there, presumably, to try to help make it. And yet, biological systems can only work. Some need left hand, some need right hand. And if they need all left-handed molecules and the right-handed are introduced, it kills the reaction.
So, all living things, as you mentioned, all living things on Earth, all use left-handed amino acids. As you can see in that picture, that's sort of what we're talking about. It's a mirror image, like the gloves. 19 of the 20 amino acids are left-handed. Glycine is not, simply because glycine is the simplest amino acid. It doesn't have a chiral image. Put a mirror in front of it, it's actually perfectly identical. So that one doesn't count. But all other 19 are all left-handed for some reason. All the sugars that all life uses are all right-handed. Now, nucleotide bases are not chiral, so their mirror image is perfectly identical, but the base attaches, as I mentioned before, to a phosphate and a ribose. A ribose is a sugar, and it's right-handed. Now, the thing about natural processes making these molecules, natural processes cannot distinguish between left and right-handed. So if you have amino acids popping up, say, they found some on meteorites and molecular clouds, these are always going to be perfectly 50-50. When the molecules go through these reactions, they don't choose which side they go on. They just go on. And since there's so many of them, it averages out to almost basically perfect 50-50. Life needs 100-0 to function. There's no known natural process that can make 100-0, only left-handed or only right-handed. The only process that can even change the 50-50 racemic mixture, which is what it's called, is a specific type of polarized light from a specific type of star. And even then, it only changes it by about 2%. We're looking at about 52-48. We need 100-0. And it's important to know that we're not just talking about, you just got to get lucky and choose the right ones in this 50-50 mixture. No. If you happen to incorporate the wrong one, it messes up the entire thing. If we as humans eat right-handed amino acids, it's toxic to us. Our body will start to incorporate them, but it won't function the same, and it will kill you. So for some reason, life needs what's called homochirality, same chiralness, if that makes sense, 100-0, and there's no known natural process to even have that happen. So, let's calculate this. Let's crunch the numbers and find the actual probability of this life form coming about. In fact, we're not even going to say the life form. We're just going to say the genetic material. We're leaving out the proteins, the lipid membrane, everything, just the genome, right? We're going to make some assumptions. We're going to assume there's an abundance of building blocks, the nucleotides, the 4 bases needed, right? A, T, C, and G. Plenty of those. We're also going to assume they're all homochiral. They're all right-handed sugars that are being used in this, right? To form the backbone. So all the same right-handedness. Not to worry about that. There's no UV damage also. Magically, somehow, this life form is not getting bombarded by UV that will disrupt its genome. And finally, most importantly, there's no mind behind this, which means there's no preference for what the next nucleotide base is. So all 4 bases each have a 1/4 chance of being chosen. We're going to assume that. So what are the numbers? Well, if you just want 10 base pairs in any specific sequence, to do the math, it's simple. It's 4 times 4 times 4, 10 times. That's about 1 in a million. So to just get 10 base pairs with 4 letters to use, it's 1 in a million to get that specific sequence. But if we bump it up to 100 base pairs, keep in mind, this is not even big enough to be a gene yet. It's not even big enough to code for a protein. 100 base pairs, we're talking about 1 in 10 to the 60th. That's 1 with 60 zeros after it. All right, the number of atoms in the universe is 10 to the 80. We're already reaching a number so high it's incomprehensible. But 100 base pairs is not enough to even make a single gene. We're talking about this thing as thousands, right? So what about 1,000? Maybe you can make one small protein, one of the 60 parts of a ribosome, right? This would be 1 in 10 to the 600. So you might say, Tim, well, we're not just having one reaction do this, right? If it's me rolling the dice on this, right, 1,000, it's going to take me to the end of the universe and even more than that to get to this. So let's assume every water molecule in the entire ocean is doing this reaction, 10 to the 44 water molecules. And say they're doing this reaction every second. Every second, it's 1,000 base pairs. Every second they're redoing it. And they're doing this for 5 million years. This is the whole window, right? Well, our chances have got to be better now, right? We have every water molecule in the ocean doing this for every second for 5 million years. The probability is 1 in 10 to the 542. The number is still incomprehensible.
So, Tim, let me say that philosophers tell us that if something has a probability of larger than 1 in about 10 to the 50th, it's never going to happen in the history of the universe. This number is
Incomprehensible.
400,
larger, yeah.
almost 500 times bigger than that.
Well, more than that. No, not 500
Oh, that's right.
times bigger. This is 500 zeros bigger.
Magnitude, orders of magnitude bigger.
There you
Yeah.
go. So it's not times 500. Yeah, it's times a number where if I tried to write out the zeros, it would go past that wall. The number is so big. Okay, so what about it's even bigger than that? What if we have every atom in the universe doing this calculation, right? Every second. And say it's not for 5 million years. Say they've been doing it since the Big Bang. 14 billion years. Every atom and every star of every galaxy in the entire universe all rolling the dice on 1,000 base pairs. That's it. Just get a specific 1,000 base pair sequence, right? Every second, every atom in the entire universe for 14 billion years. The number has to go down by a huge amount, right? It's still 1 in 10 to the 503. There's still 500 zeros. I mean, this number is so incomprehensibly large, and this is just 1,000 base pairs. What about 10,000? Now we're getting into the realm of maybe half of a ribosome, which is needed to make proteins, right? This number is now 1 in 10 to the 5,903. It just keeps getting ridiculously large. And if you want the entire genome in a specific sequence, we're talking about a number that's 1 in 10 to the 299,903. This means even if you had 1 trillion universes doing this calculation, every atom in each one for 14 billion years, you just subtract 12 from that exponent. You'd have 1 in 10 to the 299,800 and whatever, right? So it doesn't matter even if you have an almost unlimited number of universes, this genome will not come out into existence on its own. And this is just the genetic material. If you happen to have this miraculous chances complete, you have this genetic material now, within hours, it's going to start breaking down. You need a phospholipid bilayer, and you also need millions of already folded and formed proteins embedded in the membrane, inside, already creating things. This is only a small fraction of the picture, and it's already, the probability here is far past 1 in 10 to the 50th.
Wow, some great numbers, Tim. Thank you. So at this point, a cynic may be, a skeptic may be asking, but there still may be some natural process that can account for this. I want to answer that by asking you to think as a class about some of the things that you may personally create right? Tim's actually written a book. Tim's a writer. Maybe you write music. Maybe you write poetry. Maybe you knit. Maybe you cook. Maybe you invent things. But could any of those things be done by a natural process? Any process without a mind? No. Why? What's the difference? What's the difference between that and like a snowflake or a star or a crystal coming into existence, which can come into existence just by natural processes? What's in common to all of those things? And what's in common is that they each require a specific arrangements of parts. Think about when you write a book, or you write a sentence, or you text somebody. What are you doing? First, you select the letters that spell the words make the sentences make the paragraph makes the chapter make the book. And every step along the way, you're making a conscious choice. You're eliminating a whole bunch of words and including the one word that works. Think about a machine, a car, the car. Every one of those parts needs to be specifically made to a specific size out of a specific material and in a specific place and relates to the other parts that connect to it. What we're saying is that early life, that first bacteria, is just like that. You saw the complexity, you saw the machine-like character of those parts. Those kinds of systems do not come into existence on their own. They require a mind to first of all, to think it up, right? Somebody had to think of a car before they built the car. And then they had to tinker and play with it, and this doesn't work, and maybe we'll try this, and put it together using a conscious mind. And that first life form way more resembles something that's designed like us, except what we design is so much less sophisticated. Biology is so much more elegant than anything that we design. Now, there's only two ways that things get made in the entire universe. Think about it. Either things are a result of natural process, like the sun comes up and the stars are out, and things sort of fall apart as we go, and there's things that don't require a designer for them to happen. They just happen. But then there's other things that require a conscious mind to be in existence, right?
And I just want to say, you might say, oh, what about AI generating something? Yeah, there's no conscious mind for the AI, but the AI still had to be designed by a mind, right? There had to be a mind at the beginning of that. The AI didn't just come about on its own. And so even if you have, a robot making something, the robot had to be made by someone. So
the next objection may be something like this. Scientists may say, well, yeah, the origin of life is just so insanely complicated right now, we don't understand it, but I have every confidence that in the future we will understand it. You know what? That is not a scientific statement. But that's what you hear. Interpretation of data should not be based on wishful thinking. If you want to be a good scientist, you go with the evidence, and you have to live with the evidence as it is right now. And the evidence right now is overwhelming, in our opinion, that first life resembles so much more human-made systems which require a mind to put together, and yet is so much more sophisticated and complex than anything that we can build, more than something that was designed or something that was built by natural processes. So let's look at one of those quotes that we talked about at the top again. This is when maybe you read scientific literature a little bit, or you look at these science stories, and you're going to see this almost every time you read a story about origin of life. It's going to sound like this. Tim, read it again.
These simple molecules, right? They're talking about the amino acids and the sugars. combine to form more complex ones, such as fats, proteins, or nucleic acids, the DNA. Once an information system can make copies of itself, then natural selection kicks in. Once the information system can just make copies of itself, right? They're not even thinking about proteins.
There's about 100 million steps they've left out of that description, isn't there? And now you know. Now you know. Now you can be critical of articles like this. So Tim and I believe that the story in Genesis, that God created the universe, He created life. It sounds like an unsophisticated statement these days, but we believe that the evidence, not just our Christian faith, but the evidence from science agrees with that much more than a naturalistic explanation.
So a couple things we want to leave in conclusion. One here, I love this image. This is a simplified image of the metabolic pathways in a bacteria. So this would be each dot on that, it's really hard to see, each dot on that would be a change in a molecule. And this is just mapping out. A lot of this is unknown. A lot of this, a lot of these dots you can see there are unlabeled because we're still discovering a lot of things. But I hope you just look at that and you see that all that stuff is a schematic of the functions of a bacteria, not even one of us.
So in conclusion, here's what we want you to go away with. Origin of life is one of science's most difficult problems, and it's actually getting harder. You want to learn about this from a real scientist, go online and listen to a guy named James Tour. And one of the things he says is, it's like playing a football game where they move the goalpost back, right? So we make one discovery, and yet as we learn more about the cell, we don't even have a clue as to all the things it does. Every year we learn more and more and more about the complexity and the things it does. So every time, imagine a football team makes a 1-yard play and the goalpost goes back 10 yards every time they advance. That's what's happening with Origin of Life. Popular and even scientific media will misrepresent the problem. They'll make it sound like that article. They make it sound like it's simple and it's almost solved.
And not like that.
Yeah, right, the quote that we read before. Minimal complexity, simple life, you're going to hear that a lot, is about as complex as a city. It's an assembly problem. First life needed a vast array of precise environmental conditions to bring together 100 of these, 100 million parts. In just a few hours. It couldn't happen, it didn't have millions and billions of years. It had to happen from the beginning to the end in just a few hours. That car had to self-assemble from the dirt and the mud in just a few hours. There are no known natural processes that could put these parts together. It more resembles design. And life appeared in a window of about 5 million years or less. We believe that given a proper understanding of all of this, interpreting the data, design, which is a miracle of the highest order, is actually the best scientific conclusion to come to when it talks about the origin of life. And then one thing I want to close on, I talked at the beginning about the beauty of natural systems. Looking at that tree and thanking God, thanking Jesus for that tree that He made, and it's in my front yard, and all the systems that go on in it. I think when we appreciate nature, it's infinitely better when we appreciate nature knowing that there's a creator behind it who wants to please us, who wants us to have that pleasure of enjoying the beauty, and if you study a little biology, knowing the complexity of those systems. It's astounding. So we want to close actually with a verse. Tim?
Psalm 19:1. The heavens declare the glory of God. The skies proclaim the work of His hands.
So before we close, we got to say thank you to the person who put this PowerPoint together. It wasn't Tim or I, it was this guy right here. So thank you, David.
Thank you.
Thank you, everybody.
Thank you.
Yeah.
We'll take questions
Bring
now.
them on. Well, you got to get to that mic.
Test. First, we have an online question for you.
Okay.
It's from Doug McComb, and it's a,
Doug has a question? What a surprise.
yeah, what a surprise. Thank you, Doug. Let's see. He says, I am confused by what you said. Did you say that there was only a window of 5 million years when liquid water could have been formed?
You want to answer that?
How so? E.g., the dinosaurs lived like 300 million years ago. So I think he must have.
No.
Yeah, I'll clarify. So life began around 3.8 billion years ago. Earth is 4.5 billion years, so it's basically at the start of that. Dinosaurs are, a half a billion years ago. They're really recent compared to this life. And between liquid water forming, where the Earth cooled enough, that was about like 3.8 something billion years ago, and at 3.8 exactly around there, life formed. So 3.8 billion years ago, there was a 5 million year window, if that makes sense. Hope that clarifies.
So the time between when the early Earth cooled enough for life to possibly exist and the time that we know life did exist was 5 million years later. So you'll hear people say, well, the Earth has billions and billions of years for this stuff to happen. No, it actually happened in a window of 5 million years or possibly very much less. But that's the widest it can be with the evidence that we have.
Okay, here is an observation that I have for you guys. It's impossible to have computers, airplanes, cars, everything that they will work by themselves. You have to have a mechanic, a programmer. You have to have somebody that started that thing, developed it, and it's not going to work by itself. You have to have the mechanic there and everything to keep it going, right?
For man-made systems, you need a mechanic. Is that what you're saying?
You have to have somebody who designed that thing
Right.
to stay with it.
And what?
To stay with it.
Yeah.
You have to have a mechanic in your house, a programmer, and everything that you need for that thing to work. That person has to be there right next to you. Otherwise, it's impossible. Therefore, when God created, and I'm going to lose it here, when God created us and created everything, He says, for by Him were all things created. That's Colossians 1:16. Created that are in heaven, that are in earth, visible and invisible, whether there be, or there be thrones or dominions or principalities or powers, all things were created by Him and for Him. And then my thing went out here, but it continues to. And by Him, all things, and He's before things, and by Him, all things consist.
Right.
That means that mechanic, that programmer has to be in your house. So God is always in directing this because it's by His things, all things consist. He's there all the time because Proverbs 3:5 says, trust in the Lord with all your heart and He will direct your paths because He's going to be there all the time. Thank you.
Thank you, Dan. Thank you.
Should I say a comment?
Yeah.
So you mentioned, right, their mechanic needs to be there maintaining this stuff. And there is scripture that says, right, God sustains the universe. But on the cellular level, I think that's why there's such sophisticated DNA repair. This would be like if a car could give itself a fresh coat of paint and could start tightening bolts and replacing pieces in its chassis, and right? It's insane. But with this complicated machinery, I think God put that in there so He wouldn't constantly need to be tweaking with His fingers, right? This thing could do it with its insane machinery on its own. And I don't know. I see DNA repair mechanisms, and I'm just in awe of God.
with AI, so for AI to come up with the statistics you showed, it would have to. It sounds like it would be impossible to. You said AI has no conscience, so somebody has to feed something in there so it'll come up with that, which maybe is too great to ever do. I don't know.
What's your question, Eddie?
Well, can AI come up with those statistics you put up there? And wouldn't it have to be programmed to put them in?
Sure. Well, I think AI is designed to look like us, isn't it? So we look at AI and we say, wow, it looks like it's conscious. Just keep in mind that no machine is ever going to be conscious.
It'll just mimic consciousness.
It'll mimic consciousness. They will tell you this because naturalists they don't believe in the supernatural, right? So somehow the human mind and this sound and light show that we have that's called consciousness has to be explained by naturalistic processes. And if it's explained by natural processes, then we should be able to design a machine that is conscious someday. It's never going to happen because a machine cannot have the supernatural part, the soul that we have that allows us to be conscious. So it's not something you ever need to worry about. When people say that, you can just, like this. It's not going to happen. And besides, they're two completely different kinds of systems. We can prove that people are still conscious and thinking and aware when there's no brain activity. Now, if brain activity is like the electrical activity in a computer, well, if there's no electrical activity in a computer, it's not doing anything. But if there's no electrical activity in a human mind, sometimes it's still doing something. It's not even the same kind of thing. So don't ever worry about that. Don't ever be afraid of those stories. AI, it's just a really sophisticated, elegant, cool machine. But it's a machine. Jim.
Great. Guys, this is so great. My first question is, when will the writer's strike be over? That's my first question.
We'll ask AI.
But I know when Roger Penrose put together his calculations about how the probability of the universe being formed was like 10 to the 168th of the probability of the universe happening. So that's a big number. But obviously your numbers are way, way bigger. So Penrose obviously didn't take any of these. He didn't have any of these kinds of these calculations in his paradigm to put together into that. So what are your calculations are just so great just in the molecular form. So were his calculations based on just what he observed in the observable universe and not taking into account these kinds of things? And if you could come up with a number like with all the probability, would be overwhelming, obviously, because the numbers that you gave were just overwhelming. So is that what Penrose's number was based on?
Okay.
Just the universe?
Yeah.
So I believe he's saying the probability of the Big Bang happening, and this would be the probability of the origin of life happening. And so all you do was you'd add the exponents together. So you'd add his 160 to our 299,903. Yeah.
His calculations are very teeny than compared to what yours are. Because yours are magnitudes beyond what just the universe happened. I think it's just great.
Yeah, well, ours is biology is more complex than the universe, really, in some ways. Dave?
Yeah, looking at the two of you could comment. A.J. Roberts, who's a scholar at RTB, taught a few years ago a class here in Paradox about getting even more complex. And this kind of goes with what Dan was talking about of sustaining, obviously, that God continued this. That when we had these first bacteria, A.J. Roberts, I believe, you can verify that for me, said that without specifically designed viruses, those particular bacteria would multiply so much that they would have destroyed their own environment and thus not prepare Earth for humans. And in fact, another one obviously is why do we have bacteria? That's he would say, why do we have all these bacteria for so billions of years was to prepare the Earth for us. The only way that we get concentrated minerals was all these bacteria working for billions of years. And in order for those bacteria to work for billions of years to prepare planet Earth for humans, we also had to have these viruses specifically designed to control these bacteria. Am I stating that correct?
Yeah. So I studied a little bit of ecology also for my degree, and one example would be I don't know if you guys know about the wolves in Yellowstone, right? So the wolves in Yellowstone, they hunted and they killed almost all the wolves, and when the wolves went away, the deer population, which they ate, the deer population exploded, but then started to suffer from rampant disease and all this stuff, and they realized that the wolf population eating the deer kept them in check so that it also kept disease in check. And when they reintroduced wolves back to Yellowstone, the balance came back. And I think that's sort of like bacteria and the bacteriophages, which are the viruses that eat the bacteria. There needs to be balancing forces. If not, the bacteria will just steamroll through the entire Earth, which has no competitors, right, for this early life. That was probably needed for the first few million years to start changing the environment, producing, like you said, helping collect minerals, producing oxygen, things like that. But eventually, they had to be reined back in by something, and that was probably the viruses.
And if you think about it, the first life on Earth almost certainly was not one single bacteria just there in, like Manhattan Island, just randomly, because that's where God created it, or wherever. He probably created an entire ecosystem. I can imagine being there, and one moment there's nothing, and the next moment there's hillsides covered with biological systems. Because what Tim's saying and what Dave's saying is they all rely on each other to a great degree. So that makes the problem even more complex, doesn't it? And it was A.J. Roberts that gave me the analogy of a single-cell organism is about as complex as a city.
Hi. First, just a simple question, and that is the 5 million years is the window for life to start. I was wondering where that number came from.
Hugh Ross.
Hugh Ross, yeah. He came out with a paper recently where he estimated about 5 million years between when life could form and it did form.
Okay.
So, and I trust him.
The other thing I'd like to say is that in calculating all these probabilities, I don't know how the different probabilities are weighted, for example. So what if the dice are loaded in some of these places? Seems to me like, and we don't know that. So that seems to me like if you load the dice certain ways, your probability is going to be changed drastically.
Yeah.
And so that's why we should be a little bit concerned about these large probabilities. I'm not saying that the large probabilities don't exist. I mean, we understand how complex all these things are. But as far as calculating probabilities, that may be a little bit more complicated.
I'll go back to that slide. Yeah.
I want to say one thing
Please.
about that. I think calculating those kind of probabilities is impossible to do it realistically. But the point of the probabilities isn't to try to argue over, a couple of numbers. The point of the probability is to say that the odds are way, 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 way past it happening on its own, no matter what. So the numbers don't really matter. The numbers show you that it's impossible. That's what the numbers mean.
Well, we understand it to be impossible just from the general argument.
Well, impossible
that's
from a
what I'm
naturalistic
saying.
standpoint.
The numbers don't help prove it.
Say it again, Norm.
I said we understand the impossibility just from the general argument.
Yeah, and that's what the numbers really
And
mean.
the numbers don't help prove it.
You don't think so?
Well, because the numbers are not precise enough. I mean, we don't know what the assumptions. We're not aware of the assumptions that how good they are in going into calculating.
Well, okay, I disagree, but I think,
Can I answer that?
Yeah, of course.
Well, we see in nature there is actually no preference for the next base pair. If there's no proteins involved, no machinery, it is a perfect 25% chance for the next nucleotide to be chosen in the sequence. That's all I base it off of. But yeah, of course these numbers aren't exactly accurate, but I just wanted to show you that between 1,000 100 and 1,000 base pairs, just going off this calculation, we're already surpassing the number of atoms in the universe by many magnitudes. And I want you to take away 1,000 base pairs is not even enough for a gene, right? And so you can't even get a small gene coming together. That's all I wanted to illustrate with this. But yeah, of course, really hard to calculate such crazy probabilities to extreme accuracy.
Okay, hi. This is a whole change. Do you understand anything about the mRNA vaccine?
Uh-huh.
Okay, I have a question and I don't know the answer at all. I have heard, here's it, okay, that when you get the mRNA vaccine, it has been shown to travel through your body into your liver where they were tracking it, and in the liver to enter the nucleus, and in the nucleus to alter the DNA, which they said couldn't happen, but some people have said, oh, look at this. And when it does that, it makes spike protein your friend rather than your enemy, and therein causes great damage to your cells. Could you comment on that?
Sure.
I'll let you do that.
Yeah, sure. So the mRNA vaccine, mRNA, well, I'll start with this. The central dogma of biology is DNA makes RNA, makes proteins. The DNA stays in the nucleus. This is not bacteria we're talking about, this is us. The DNA stays in the nucleus, or where the bacteria would just be the chromosome, right? But we have this wrapped up nucleus that keeps it all tidy and safe. Then, I don't know if you remember that picture of the crazy colorful thing, right? These gated channels allow the mRNA that's inscribed from the DNA to leave the nucleus and now enter the body of the cell. Here, the mRNA starts making proteins. Now, the thing with RNA, any RNA in our body, including mRNA, messenger RNA, it's called, is that it breaks down really fast. Within hours, if not days, if not hours, the RNA is broken down very quickly. If it wasn't, anytime we make RNA to make proteins, it would produce too many proteins. It would stay around in the cell, ribosomes would attach to it, start building too many proteins, and it would clog up our machinery. So mRNA doesn't last too long in the body. So after you get the mRNA vaccine, within a day, all that mRNA will be broken down. I don't think it'll even make it into the nucleus. To make it into the nucleus, you would have to go swim upstream through those gated channels. And the way the nucleus is designed, it's designed so that it's basically one way. RNA is not supposed to go back into the nucleus. So I think it is improbable that the mRNA vaccine would somehow go into our DNA, because RNA can't be incorporated into DNA. It's a different step, it's a different molecule. If you try and put RNA nucleotides into DNA, it'll be rejected, it'll not function. So just from my knowledge, I think it is extremely unlikely that the mRNA can be incorporated into our genome. That's my answer.
Okay. We've got a little more time. One thing about AI, I think the fear is not consciousness of AI, but rather giving authority to AI. That's the fear now, that humans would give up their authority and AI would make that, a billion times faster than we can think, with far more information and make decisions for us. Chat AI, you probably heard of that, and it's already doing that. So that's the fear of it. I think Doug's got another question here. He says, there is also the added improbability of biological life simultaneously having food sources emerge alongside them. That should add to the exponent. That's kind of like the chicken and egg thing. Each part is dependent upon the other. Do you want to say anything on that?
Yeah, sure. I mean,
Go ahead.
there's all these chicken and egg problems, right? You have the DNA, can't exist without the proteins, but both of those have to have the sugar, the ribose to form the backbone, and to feed this thing.
And the ribose can't come into existence.
That's the thing. There's a lot of research that's been put into how can ribose come about naturally. There's no answer so far. It's been looked in for 60 years now, and nobody can find out how ribose can be made naturally without an organism. And even if it is made, as we learned, it'll be made in a 50-50 mixture, left and right-handed, which is trash. That can't be used by living life forms. It needs to be 100-0. So, right, every time you even bring in, what about that sugar? You're basically, you might as well be doubling that exponent. I mean, right? It's crazy.
Okay. Well, guys, it's about time to quit. And it's been great. Really appreciate it.
Thank you.
So, which one of you would like to close us in a prayer?
Sure. You mind if I do?
No? Go ahead.
God, Father, thank you so much for this beautiful universe. Thank you for filling it with an array of beautiful, complex organisms. God, please help us keep in mind your love, most of all, that when we leave this room, we not only take with us scientific facts, but we take with us an appreciation for the beauty that you've created and the love you have for us, God. Please fill us with your Holy Spirit and allow us to be more compassionate, more empathetic to other people who don't have scientific backgrounds, don't know about these things, because we're all learning, God. And with your help, we can learn a little more, and we can be more loving followers of you, Jesus. We pray this in your name. Amen.
Amen. Thank you, everybody. Let me know if you need some rice.